We have seen this flowchart before when we were looking at the intro to metabolism and our carbs anabolic process, so the building up. We looked at how glyceraldehyde 3-phosphate was the 3-carbon compound that came out of the Calvin cycle in our plants during photosynthesis. And then from there is the base structure to form our major macromolecules. We then looked at that gluconeogenesis was that process of taking small 3-carbon compounds and building them up into glucose. Glucose is that primary monosaccharide that then builds up our polysaccharides. When a plant or an animal has enough glucose to run its cellular processes through cell respiration, the rest needs to be stored. We are focusing on the human side, so looking at what's called glycogenesis. Glyco is the process of making glycogen. Genesis is the beginning, the formation of. Therefore, when we have excess blood glucose, our cells are at high cell respiration rates. We're not requiring more ATP production, or in terms of people who have type 2 diabetes, they may not be sensitive to the insulin hormone anymore to start that cell communication pathway to uptake more glucose. There's a lot of parameters that can go on here. Essentially, we have enough glucose, therefore we're going to form glycogen. This is where we're then going to store, so we can store glycogen in our muscle cells, or in our muscle tissue, and in our liver. Too much glycogen, it will get converted to fatty acids, and that's where we get like fatty acid liver disease. We will store this glycogen until we need it in future use. Our body will regulate and track our glucose levels, but we can also have it measured when we have a blood draw. What we look for within that normal range of glucose is between 70 and 110 milligrams per deciliter. If a person has below normal glucose levels, they can have a condition called hypoglycemia, which means that they are not maintaining enough glucose within their bloodstream. If one has blood glucose levels consistently above normal levels, this is considered hyperglycemia. People with type 2 diabetes tend to fall under this classification of hyperglycemia. If diabetes goes undiagnosed for long term, that person can start secreting glucose in their urine. This is not what's supposed to happen. Within the nephron of the kidney and the filtration, glucose is not supposed to make it past the loop of Henle, really not past the glomerulus bundle that is in that nephron. So the presence of glucose in urine is a red flag. If someone does have glucose in the urine, this is called glycosuria, or sugars in the urine. What the body does to help regulate our blood glucose. The two very important organs are our pancreas and our liver. These have the central role in the regulation of blood glucose concentration. The cells in the pancreas will secrete hormones, which will then alert and tell the cells to either uptake glucose or not to take it up. Hormones are chemical signals. Glucagon and insulin in particular are protein hormones. So they come from the macromolecules within the protein category. If you are fasting or you are exercising, and you don't readily have glucose coming in from digestion, we need to be able to access glucose on demand. This is what's really important about functions within the brain as well, because it needs a high concentration of glucose to run all of the neurons all the time. Glucose can be broken down from glycogen within the liver. So the pancreas, with its alpha cells, will secrete glucagon. Glucagon will then travel to the liver, and it signals the enzymes to break glycogen into glucose, sending glucose out into the blood so that it can then be used by those cells. As we raise that blood sugar and as we raise the concentration of blood sugar, we're going to have a signal sent to the pancreas that we now need insulin. Insulin is then going to be released. Insulin will then tell our tissue cells it is time to uptake all this glucose that the liver has dumped into our bloodstream. Insulin comes from our beta cells within the pancreas. Here, this is an image of the protein of insulin. The first image, because we have not done proteins yet, this is called a space filling model, kind of gives you the three-dimensional orientation of this protein. The one next to it is called the ribbon model. It shows us where specific folding is occurring, gives us more information about active sites and interactions that can occur. People with type 1 diabetes typically have destroyed beta cells, so their lack of insulin production is the reason why they are typically required to take in injections of insulin. If there is enough blood glucose, insulin will also be sent to the liver, and that will then signal glyconeogenesis to reform glycogen. That will then also result in lowering of that blood sugar. So all depending on what we need to do, we have these chemical signals. Glucagon breaking down glycogen into glucose, insulin telling our tissue cells to uptake glucose, or our liver cells to convert glucose back into glycogen. When the cells are ready to uptake our glucose, what happens is insulin has to start by binding to a receptor on our cell membrane. That's going to release a cascade pathway where there's a lot of different enzymes and chemical structures that are going to come together, essentially sending chemical signals. One of those chemical signals is to activate our glucose transport protein. There are a couple different transport proteins. I'm not testing you on that. That is totally physiology's job. But if we need to uptake glucose, this protein gets activated, allowing glucose to come in. Once glucose is in the cell, a couple different pathways can occur depending on the cell it entered. It can undergo cell respiration, that's where we're going to get our ATP. If we have too much blood glucose and we're working with the liver cells or muscle cells, we might signal the production of our glycogen. Or if we are building structural or we have just too much glycogen, we can also signal the formation of our fatty acids. The cells are constantly communicating of what is the needs and max efficiency, so the glucose knows where it needs to be shuttled based off the cell it went into and the hormones that communicated what we need. The human body is a very complex system. Diabetes can occur based off our ability to regulate blood glucose. We have type 1 and type 2. Type 1 diabetes, the cause is relatively unknown and there's active research occurring within this field. However, we do have some theories. One theory, and really the reigning theory right now, is that the body's immune system is going to be destroying the insulin producing, or what people will call islet cells. These are the beta cells I just talked about. That destruction, that's more of an autoimmune disorder where our immune system's attacking our cells. It can be tied to genetics. There's a theory that there are certain viruses that can mimic the disease, and that will then trigger that immune response and can attack the beta cells. The least researched right now is exposure to environmental factors, whether this is to health and diet or chemical exposure to what we have in nature. Because as we know, some of our chemical compounds do not readily degrade in nature, and a lot of them can mimic certain hormones. We just don't have enough information about our exposure to some of these synthetically made chemical compounds and what effects they may have in terms of diabetes. Type 2 diabetes, we do have a lot more understanding of how this is caused. What occurs is either the body does not produce enough insulin, or more commonly, what we see is that we have a built-up insulin resistance. That means the receptors on the cells are not as sensitive, so when insulin goes in and binds with its receptor, that chemical cascade pathway does not get triggered. Therefore, the glucose proteins do not open to allow transport to come in. Some people are more prone to diabetes based on their genetics and their social history as well.